Hey everybody, I want to do a lab for you guys today, so you can follow along in this video, and the instructions I've posted. So this is the genetic drift of M&M's lab, and I'm just going to go through the procedure and explain what I'm doing here for you, and you can record the data in your tables. So you'll notice on the last sheet, the very last page, there's three data tables here where you're going to record your information that we're going to do right now. And then there's some other data tables in earlier pages where you're going to process this data. So the back page is where you want to focus right now for recording your information. But we're going to look at two different phenomena today that influence the frequency of alleles in populations. So first one is founder effect, and then the other one is bottleneck effect. So they're similar but a little bit different from each other, and we'll just get a sense of what each of them are and how they work by doing this lab. So at this point I'm set up with two cups. I have 40 M&Ms in population A and another 40 M&Ms in population B. So hopefully you had a chance to read through the background and the procedure before starting the video. But at this point, what we're going to do is start with the initial population heading, and I've picked my 40 M&Ms for population A. So we're just going to scatter them on the napkin so you can see them better here. And basically, I'll just sort them into color, and you'll be able to count them up and record them in your data table. So again, this is population A. Okay, so hopefully you're able to see the numbers of each of the colors here. And so you're going to write this data down on the last page in table 1. So you'll see that it has a column, population A, numbers of alleles, and frequency of alleles. So the number is the actual count. So we go from blue to green down to brown. So the number of alleles for blue is 1, 2, 3, 4, 5, 6, 7. And so you can just count up the other numbers and put them in the number of alleles column. And then beside that is frequency of alleles as a percentage. So there are 40 M&Ms here. So if you wanted to know the frequency of alleles for blue, we would take 7 divided by 40 in total, and then multiply that by 100. And let's figure out what that is. My brain can't do that on its own. It is 17.5%. So that would be your blue frequency percentage. And so you can do the same for all the other colors here. So that's population A for table 1. And now I will organize population B. Okay, population B all set. And again, I just randomly pulled 40 of these out of my M&M bag here for both populations. So it's completely random which alleles I picked out for each population. They're probably going to be a little different. So in table 1, you're now going to write the number of alleles for population B. So again, blue this time has 5, and you can go down and fill in each color. Frequency of alleles, same thing, 5 blue divided by 40 would be, what is that going to be, 12.5%. And you can work out the percentages for each one there. So that's table 1 all filled in. And now we have taken care of steps 1, 2, and 3 in the initial population section. Now we're going to move on to apply the founder effect on these populations. So what I'm going to do is put all of these back into their cups. Okay, and I'm going to mix them up randomly. I will not look. Okay, so first step is, without looking, take out 8 M&Ms from the population A cup and place them on the paper towel. This is your founding group. So this represents the entire population. And remember that these are alleles. So each organism is going to have 2 alleles. That means if there's 40 alleles, this actually represents 20 organisms, whatever they happen to be, the M&M organisms. So I'm going to take out 8 random M&Ms here, and the founder effect is when part of the population gets relocated into a new area and it becomes separated from the original population. So we'll see later on in our analysis if that has any impact on the percentage of the alleles. So 1, 2, 3, 4, 5, 6, 7, 8. Okay, so now what you want to do is go to table 2, the middle table on the last sheet, and for population A, we're going to write down all of the colors that we have in our founder population. So blue, there is 1. So in your number of alleles, you would put 1 for blue, and you'd go down the list. We don't actually have all the colors, so green would be a 0, and yellow would be a 0. And we've got, what do we have? 3 brown, 3 red, an orange, and a blue. And so at this point, you can fill in the frequency of alleles as a percentage and do it the same way, except this time you're dividing by 8 because there's only 8 here. So blue would be 1 divided by 8, 12.5%. Orange would be, again, 12.5%. And then you can do the red and the brown. Of course, the ones that are missing are going to be 0%. So that's the founding group. Let me just write that down. This is the founding group for A. And this is going to be the founding for B. And I will take out 8 from B. Okay, there's my 8 in the founding group for population B, and we'll just organize these. Okay, so you can fill in table 2, the population B founding group. No blue, 2 green, 2 yellow, an orange, a red, and 2 brown. And again, turn that into percentages for population B. And that will round out your data table number 2 for the founding groups. And that is the founder effect. So we've gone through step 1 and 2 there. That's all done. And let's see here. We're going to move on to bottleneck effect now. So I will put all of these back in to our original population of 40 alleles. And we're going to do, this is a totally different simulation here. So we're going to start with our original populations again, and this time we're going to look at an effect called the bottleneck effect. So what we're going to do is remove 30 of the M&Ms from population A cup. So let's say a natural disaster came around this area where the population was living, and it wiped out most of the organisms, but only 5 of them were left. So that would mean there will be 10 M&M alleles left in the cup, and 30 alleles would be wiped out because the other 15 organisms were wiped out. So we're going to have 10 alleles left. So I'm going to take out 30 of those, and I'll just put them over here. That was a big hurricane. I don't know if you could hear the wind in this classroom, but the windows have all been blown out and the desks are all knocked over. It was a very strong hurricane. We lost 15 out of our 20 M&M species, and we now have only 5 individuals left. So let's take a look at the alleles that are remaining in the population. So in your data table number 3 on the last page, you can now fill in the surviving population after the bottleneck effect. So the hurricane came through, and this is what we have left in the population. So 1 blue, 1 green, 2 yellow, 4 reds, 2 orange, and no browns, sadly. So when you're converting that to the frequency of alleles as a percent, this is easy because it's just divided by 10. So 4 to 10 is 40%, 20%, 10, 10, 20%. Easy calculations for table 3. I will now have, let's say, what's going to come through this time? Tornado. Tornado's going to come by, and we're going to have population B. Many of them will be wiped out. So we're going to lose 30 here. There we go. And only 10 alleles left in the population B now after that tornado. I think my hair got messed up a little bit there. Kept my clothes though, so that's good. Alright, we have 3 orange, 3 blue, 1 red, and 3 brown for the bottleneck population for population B. And so that will fill in the right side of your table 3, and percentages, of course, 30, 30, 10, and 30 for those. So that is it for collecting the raw data for original populations after the founder effect and after the bottleneck effect. So at this point, you are moving into the discussion questions, and so you'll have access to the Google Doc, your own copy of that, and you can just type in the answers in there and use as much space as you want, type as much as you want. If things get shifted around, that's fine. So to clarify what you want to do for specifically question number 2 and number 5 here, on the discussion part, you have 2 more data tables, and let's just focus on question 2 here. So you're basically just pulling the data that you wrote down on your last page and putting it into these tables so you can compare it more easily. So this first row here is the allele frequency in the original population A. So that's this second column here for population A of the initial population. Just take those percentages and write them straight across the first row here for each of the colors. And then you can do the same thing in the third row, the original population percentages for population B from the first table. But in the second row and the fourth row, you're going to write in the allele frequency in the founding population of A and the founding population of B. And that would be in table 2. So you're going to be writing the percentages in this column here and this column here in that data table for question number 2. And so it'll allow you to easily compare how the percentages of each color have changed. And then you'll do the same thing. Basically, row 1 and row 3 for that table are going to be exactly the same in number 5 because they're looking for the original population percentages of A and B in there as well. But for row 2 and 4 in that table, you're going to pull from table 3 because this is for the bottleneck effect. So you'll take the percentages in this column here for A and that column here for B and just pop them in to these rows in that table there. And you'll be able to answer the questions based on that data. Okay, so moving on, we have basically just written answers for the rest of the questions. Except when you get to number 12, I want to clarify how to do this data table. It's a little tricky if you're not used to these types of calculations, but what we want to look at is the percent change of each of the allele colors before and after the phenomenon. So the percent change after the founder effect, population A, we have to figure out the percent change for all of the 6 colors. So the equation to use is right above the percent change is the allele frequency after the event, take away the allele frequency of the initial population, and then divide that by the allele frequency of the initial population, and then multiply by 100 to change it to a percent. So where are all of these numbers? Well, you can go back to your data table, your 3 data tables on the last page. So if I was going to fill in that first box here for blue, the percent change in the founder effect population A, this is what I would do. I would find the population of blue in the initial population here. That would be that top box there as a percentage, and I would be comparing it to the allele frequency after the founder effect occurred for blue in data table 2, that percentage there. So those are the 2 that you're going to be putting at the top. So after the event would be that percentage there in table 2, and subtract the initial up here, and then divide by the initial. So again, divide by that percentage in table 1. You'll get a decimal number, multiply it by 100, and that will be the value that you write in here for blue in that first box. So you would continue that process looking at all of the colors and doing that for population A for founder effect, then population B for founder effect, and then you're going to be looking at comparing table 1 and table 3 because you're comparing the initial to the bottleneck effect for population A and the initial to the bottleneck for population B. And these are all going to be recorded as percentages. Some might go up. The percentage frequency might go up for different alleles, and it might go down for others. So some of these percentages will be positive and some will be negative. That's normal, so don't worry about that. That just means after the event happened. If it's a negative percent change, then the frequency of that allele went down. And if it's positive, then we actually have a higher percentage. So my guess is the 40% red here, 4 out of 10, is going to be an increase in the frequency of red compared to the initial population. But then there will be others that have a big decrease, like the ones that don't even exist after. If that color is gone, then that allele is gone. It's going to be a negative percent change. So that's how you would do table 12. And 13 is the question where you take that data and make 2 graphs. And I'm going to make a separate video to show you how to use Google Sheets, because everybody has access to Google Sheets. And it's actually really quick once you get your numbers typed in the spreadsheet. I'll make a quick video and show you exactly how to take that data. With a few clicks, you'll have your graphs ready to go. And then you can actually cut and paste those right into your Google Doc for the lab report. So it's pretty straightforward if you see the video on how to do it. Even if you haven't done much graphing before, just follow along in that video and you'll be fine. So that's the lab, and it won't be due until after March break. So if you want to spend time on it during March break, you can, but it won't be required to hand in until the week after March break is done. So that's all for today, and I have a lot of M&Ms to eat, so I have to go. I'll see ya. Talk to you soon.